Today I'm going to try to answer two questions, or two sets of questions. The first one has to do with one I've been receiving all my life from my friends and from my family, and that is, where do you go when you're not at UCLA, and what do you do when you're there, and how come you're never around, and where are you, and what are you doing? That kind of thing. The second kind of question is, what is a nice boy like you doing studying frogs? Well, the second question I think is easier to answer than the first one, so I'll start with that. The best answer I've ever seen written to answer that question was by Carl Linnaeus, the famous Swedish physician and inventor of the system of binomial nomenclature that we use today. He said about amphibians that these foul and loathsome animals are abhorrent. Can I have the lights down, please? Are abhorrent because of their cold body, pale color, cartilaginous skeleton, filthy skin, fierce aspect, calculating eye, even more down, offensive smell, harsh voice, that's my particular favorite, squalid habitation, and terrible venom. And so their creator has not exerted his powers to make many of them. I can't think of a better reason to study frogs than that. Now, another man who influenced my life, beside Linnaeus, was August Krogh, the famous Danish physiologist who received the Nobel Prize in 1920 for his pioneering work on capillaries and capillary action. He said, among other things, that for a large number of problems, there will be some animal of choice on which it can be most conveniently studied. Or in other words, organisms that exhibit extremes of adaptation may reveal general principles not readily observable in less extreme species. And it's this last statement that really forms the guiding principle, now known as the Krogh principle, in fact, for neuroethologists or physioethologists. I am a neuroethologist, meaning I study the neural basis of behavior, or physioethologist, studying the physiological basis of behavior. Well, the person who actually taught me about the Krogh principle expressed here was not Krogh himself, but rather my thesis advisor at Cornell, Robert Kapranica. Bob, actually, these are some pictures I took of Bob in Puerto Rico when he accompanied me on my very first overseas research expedition in the 1970s when I was doing my PhD thesis. And Bob did some pioneering work in neuroethology, and I want to talk to you now about one experiment that he did that forms a kind of model or paradigm for many, many studies in the field. He worked with the North American bullfrog, shown here. On the left is the male, on the right is the female, and you can tell the difference between males and female bullfrogs because the eardrum of the male is much wider, has a greater diameter than the diameter of the eye, the male, where it's about the same size in the female. Would that this be true for all 5,000 species of amphibia? It is not. Now what Bob did was he synthesized the call of the male bullfrog, shown in the next slide here. Oh, by the way, I want to point out that this is the tympanic membrane I was talking about. That's the eardrums, or tympanic membrane, shown here. He synthesized the call of this bullfrog, and this is shown in the top portion of this slide, where you can see the synthetic bullfrog call. You notice it's a pulsatile call containing, in this case, 6 pulses, and if you expand the time axis in the middle of the picture, you can see that it's a periodic call with a period of about 10 milliseconds. And down below, he's showing an amplitude spectrum. This is work from Kapranica. He's showing an amplitude spectrum in this picture, which plots amplitude on the vertical axis and frequency on the horizontal axis, shows you at a glance all of the frequencies contained in the bullfrog's mating call. And as you can see, in this call, the call consists of 2 bands of energy, a sharply tuned low-frequency band centered at about 200 hertz, and a more broadly tuned higher frequency band centered at about 1400 hertz. So what Kapranica did with this synthetic call is he went into the laboratory and he took a loudspeaker and he played back this synthetic call to an isolated male bullfrog in a cage in his laboratory. And he observed the behavior of the male bullfrog. And what he found was every time he played back this synthetic call, the bullfrog in the cage heard the call and emitted his own mating call. So Kapranica termed this behavior the evoked vocal response of the male bullfrog, or the EVR. So here's a graph showing the percentage of responses and the kind of call he played back. With the synthetic call, you got 100% response every time the bullfrog responded. Now Kapranica did something which was unprecedented at the time. He electronically modified the call of the bullfrog by 2 ways. First, he low-pass filtered the call, throwing away the high-frequency energy, and playing back just this low-frequency piece to the frog. The frog didn't like this. He gave no responses whatsoever. Okay, you could tell the difference. Then he went back to the original call, and he filtered it the other way. He high-pass filtered, throwing away the low-frequency energy. And again, the bullfrog's response with the high-pass filtered call was 0. So from this very simple yet elegant set of experiments, Copernicus was able to conclude that both the low-frequency and the high-frequency peaks must be present simultaneously in the stimulus to obtain the evoked vocal response from the male bullfrog. I spent these few minutes telling you about Copernicus' experiment because it's going to form a paradigm for many other experiments that are done, not only my own, but many people in the field. So I'm going to tell you today two stories, or two different types of experiments that we've been doing over the years with my students and my colleagues around the world. And we're trying to tell you the way we operate both in the field and in the lab. The first of these experiments is called Crossmodal Integration in a New World Frog. Well, crossmodal integration, we can ask the first question, and that is, what does this mean? Well, crossmodal integration refers to the joining together of two cues of two different modalities. In this case, we're talking about auditory and visual, into a unified percept. And I can give you an example in the human literature known as the McGurk effect. Anyway, what we found is that in the Amazonian dendrobatid frog, Allobates femoralis, this cross-modal integration that I'm talking about leads to aggressive or fighting behavior among males. So we can ask two questions about this fighting behavior. We can ask first, what are the particular features of the auditory and the visual cues that lead to fighting among males? And second, is it possible to experimentally evoke aggressive behavior in a frog, which had never heretofore been done? Well, these animals, Allobates femoralis, they live in northern South America in a place called French Guiana. And in order to study them, I have to fly from Los Angeles to Miami, spend overnight in Miami, the next morning up early, take an Air France flight from Miami, stopping in Haiti, Guadeloupe, Martinique, and at the end of the day, Cayenne. Now, it's a long trip, but we spend the first 2 days in Cayenne buying our provisions, our food, our hammocks, our mosquito nets, etc., and basically getting ready for our trip. Now, maybe some of you know this, but French Guiana is actually not a country. It's what we call a département d'outre-mer de la France, an overseas department of France. This is France, it just happens to be in South America. These people are French citizens, they speak French, they use the euro, and in fact, if you pick up the phone in Cayenne and call Paris, it's a local call. If you call Brazil, which is 40 kilometers away, it costs $1 million because that's go through Paris and then back to Brazil. That's the way it is. And anyway, so when we get all our provisions, we load them all up in a van and we hire a driver, and he takes us to the end of the road in Regina, where we offload everything into a pirogue, which is a motorized canoe, and we put everything in the canoe and we go up the river, the Apelrag River, for 5.5 hours to the field station built by the French at Aratai. Now, when we go up the river, this is shown here, this is our pirogue and going upriver, it's absolutely pristine. None of the large rivers in French Guiana are navigable by large ships because of the rocks. In fact, in order to get to the field station, we actually have to go up 9 waterfalls. But we hire a man called Monsieur Meade, who knows every rock in this river, and it's very easy with him. He's worth his weight in gold, taking us up there, which is approximately what he charges us for the trip. When we get up there, we're at Aratai, and here we have, this is a place which is a lowland, wet, tropical forest. It's a beautiful spot. Outside Central Africa, it has the highest diversity of amphibians I've ever seen. And it's a place which is ideal for studying frog and frog behavior. This is our camp. It contains 3 buildings. This is a dining room and kitchen shown here, our laboratory over here, and if we could have the lights down just a little more, please. A carbe on the left, which is a sleeping platform. It's a raised wooden platform covered by a thatched roof and no walls at all. So what you do is you hang your hammocks from the rafters inside, cover it with a mosquito net, and you're good for 3 weeks. Now I've discovered that the French are incredibly civilized. And what I mean by that is they figured out how to take the water, this dirty water from the Aratai Creek, and filter it using charcoal, and storing it in storage tanks, and then running it with pipes to the tap. So we routinely drink the tap water in this field station and never get sick. None of us has ever gotten sick. It's a beautiful system. Secondly, they figured out how to have power 24/7 without a noisy Honda generator making smoke and ruining our recordings. Instead, they have large solar panels which charge batteries and capacitors, so you have silent power day and night. which is nice because it enables us to have a kitchen full of freezer and a refrigerator for our food. It's very, very comfortable. But the most civilized thing that the French have figured out how to do is they figured out how to have diurnal frogs. Most places where I work, the frogs are nocturnal. We go over there, we have dinner, and then we take all our gear and we go into the forest, and we work from about 8 o'clock till about 3 o'clock in the morning when the frogs stop calling. Not in France. In France, the frogs call at daytime or after lunch, and they call until the sun sets, and then they stop calling, and we go back to our field station and have a French meal with French wine. It's very civilized. So this is what we do. We go here and we work on these frogs, and this is the frog we came to study, the Elevates femoralis. It produces a 4-note call, advertisement call. You can see it here. Notice its vocal sac moving 4 times every time it makes a note. 1, 2, 3, 4, 1, 2, 3, 4, like that. Okay? So if we look at this call, we can synthesize it like the Pranica did, and we synthesized it, and this is shown in this particular slide here. What you can see in this slide is called a sound spectrogram, which has frequency on the vertical axis and time on the horizontal axis. And you can see here the 4 notes, 1, 2, 3, 4, that's a call. And we synthesized 10 of these calls in a row, and the 10 calls we term about, and that lasts roughly 8 seconds. Following a bout of 10 calls, there's an interbout interval of about another 8 seconds here, and then this bout and interbout interval are repeated 25 times for a total of about 400 seconds, and this is called a trial, 1 trial. It takes 400 seconds. So what do? Well, let me show you a little bit about the actual behavior of the animal in the field. Here we have, in the field, we did this, we had 2 loudspeakers and a frog between them. And what we're going to do now is activate 1 speaker at a time, and I want you to see what happens. Oh, sorry about that. Okay. Left speaker is activated. Frog goes over to investigate, thinking that there's a frog in his territory. Now that's turned off, we activate the right one. These frogs are amazingly territorial. They will physically and vigorously defend their territories against intruders, non-specific intruders. And you can play this game all day. You can now activate the left one, they'll go back and forth. They never get tired, they never adapt. They just want to kill the frog that's in the territory. Okay? But many female frogs and males of some species, like this one you just saw, exhibit what we call positive phonotaxis. That is, they're attracted to a sound. In fact, in this case, to a loudspeaker broadcasting their conspecific mating call. However, as far as we know, female frogs do not mate with loudspeakers, and male frogs do not fight with loudspeakers. And it's this last statement that I'd like to explore today. In order to evoke aggressive behavior, it's never been done because people use loudspeakers to try to evoke this behavior. They don't attack loudspeakers. They investigate them, but they don't fight with them. So what we did in order to avoid this problem was we built an electromechanical model, or robot, which we used to present the stimuli to these frogs. And this consists of what we call the robo-rana. It's a life-size model of the frog. It's hard to see, but I'll show you in a moment. Sitting on an artificial log, inside which are the electronics that run the sound system and the hydraulics that inflate and deflate the vocal sac. And this is all connected to a control box by a 7-meter cable, which is run by the experimenter, i.e. me. And then we have another piece of equipment which we call an external loudspeaker, which is also capable of producing sounds, and I'll show you how that works in a moment. If we look at this up close, we can see the robo-rana sitting on a turntable. So the turntable moves back and forth and simulates a live frog. 2 centimeters behind the model, we have a loudspeaker in this branch, which produces the sound. You see the inflatable vocal sac, and then you see a real male, the Elevates femoralis on the right here, actually attacking the model in this drawing. So here's what we did. We take our artificial log, we place it down in the forest during the day, 2 meters away from an actively calling male in his territory, and we activate this log, and we film the behavior of the territorial male. And here's what we found. This is a summary of all our results, just combined into one picture for you. So what we have here is the following. When we activate the log using sound only, this is what we call a unimodal control. Only acoustic stimulus coming out of this log, no vocal sac motion. Sound only, What happens? The male sitting in his territory hears the sound, he stops calling, he orients toward the log, he goes hop, 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 jumps up on the log, comes right over to the model, looks at it, but never contacts it and never attacks it. Okay, ever. So the sound does not induce aggressive behavior. Positive phonotaxis, yes. Aggressive behavior, no. The next unimodal control, we have the vocal sac out and pulsating, moving, but no sound. Again, the animal never contacts the model and never attacks the model in these conditions. Now the next several stimuli are what we call bimodal stimuli. Here we have the sound on, and the vocal sac in this first case is in, which means it's inside the animal, not moving, and again, no contact, no attack. In the next row we have the sound on, the calls being produced, and the vocal sac is inflated, it's out, but it's stationary, it's not moving, it's not pulsating. So this is what we call a static visual cue, plus the sound, and in this case we saw 11% of the contacts and no attacks at all. And finally, of course, the last situation, we have the sound on, and coupled to this is the pulsating vocal sac. So this is really a mimic of the real frog, okay? Using our model to mimic the real frog. Here we saw 89% of the contacts with the model and 100% of the attacks. And I'd like to show you one of those attacks in this next video. We have the lights off, please. Here we have the video showing the male attacking this model in his territory. This is the first time that anyone is able to evoke aggressive behavior in any amphibian under controlled conditions. So that was the interval interval, and notice there was no motion of the frog during the interval interval. Now here come the 10 calls. He's unhappy. Interval interval, no motion. Now we actually had with us 5 replacement models, a total of 6 models. This is one. We had 5 others in case something happened to them. We had to be able to replace them. Well, in fact, out of the 6 that we had, 2 were destroyed by these frogs. We built this vocal sac out of a condom, and in two occasions, the territorial frog put his arm right through the condom. I just hate when that happens. So what do we learn from this? We learn that the results of these experiments support the hypothesis that aggressive behavior in this species is more likely to be evoked when acoustic cues are coupled, that is, presented simultaneously with dynamic visual cues, than when stimuli of either modality are presented alone. Let the lights up a bit, please. So the idea is that when we couple these 2 cues, the visual and the auditory cue, that's when we get the aggressive behavior. But this coupling is very interesting. We can ask another question now. We can say, how strong must this coupling be to evoke this behavior? And to answer this question, you can do 2 kinds of experiments. The first we call temporal unbinding experiments, and the second we call spatial unbinding experiments. We did both of these type of experiments, but I'm only going to be able to talk to you today about the spatial unbinding experiments. Okay, so what is a spatial unbinding experiment? In this case, what we do is we present an acoustic cue to the animal. That's the stimulus, the call, right here. Present this from the speaker. And we also simultaneously present a visual stimulus, which is the vocal sac, from the model. We present them simultaneously in time, but we separate them in space by a distance d. And this distance d is variable. So here's what I mean. Here we have a frog down below, which is in his territory, in French Guiana, sitting and calling, unaware that we're even there. We place down in the forest, 2 meters away from him, our roborana right here, the model, and also 2 meters away this side, an external loudspeaker. And we separate these 2 devices by 50 centimeters. And what we're going to do now is present an acoustic cue from the speaker and a visual cue that is the vocal sac motion from the model. And we're going to ask the frog, what do you do? And let's watch what happens. Okay, there are the 2 cues. He goes over to the loudspeaker, and he jumps on the loudspeaker and looks in. Okay, that's all he does. Now we change the distance between the 2 cues, and we repeat the experiment. The 2 cues simultaneously. He goes to the loudspeaker at 25 centimeters. Now when we change the distance between the 2 cues to 12 centimeters, we repeat the experiment. Now he attacks the model. Why? Because he's doing the following. He's assigning the acoustic cue coming from here to the position of the visual cue coming from here. This is what we call cross-modal spatial integration. He's coupling the 2 cues. He thinks now there's a real frog in his territory, and he fights with it. When we repeat it for a 2 centimeter separation, the same thing happens. He fights. So he's able to do this cross-modal spatial integration over a distance of up to 12 centimeters. So in summary, if the distance between the two types of cues is 25 or 50 centimeters, he investigates the sound, because the sound is the long-distance attractor. If the distance between the two cues is 12 or 2 centimeters, he does cross-modal integration, and he fights with the model because he thinks now there's a real frog in his territory. So what have we learned from all of this? We've learned that mechanisms by which the brain binds together inputs from separate modalities to effect a unified percept of events are poorly understood. And that cross-modal temporal integration, which I didn't show you, and cross-modal spatial integration, which I did, are associated with aggressive behavior in this species. And that this system has the potential for the study of cross-modal integration, both behaviorally and neurophysiologically, topics that are currently receiving increased attention. In fact, a few weeks ago, Melissa Coates in my laboratory and I received a supplement to my NIH grant to study the neural basis of this behavior at UCLA. And we plan on doing that very soon. Now, this cross-modal integration, I should tell you, is very much like what we all do when we go to the cinema when we watch television. The actors on the screen are walking around the screen and they're moving their lips, and then it looks like they're talking, but the speaker is fixed. The speaker isn't following the actors. So what we're able to do is take the auditory cue from the speaker and assign it to the visual cue on the screen and get the illusion that the actors are actually talking. We can do that over a certain distance. But this is the first demonstration that an animal in its natural habitat, with a non-invasive technique, also exhibits cross-modal spatial integration. And that's why it was unique. So I'd like to thank the people who work with us, especially Dr. Walter Höhl from the University of Vienna in Austria, my long-term collaborator, and his student, Daniela Grabo, and several other people there, including some random biologists over here. Okay, that's the end of the first experiment that I want to tell you about. So I'd like to end the talk with just a few more slides, talking about something I mentioned earlier. I said, in fact, there are 2 frogs that we know of in the world out of the 5,000 species that have this morphology of the recessed eardrum. One lives in China, I mentioned, and one lives in Borneo. So last year, my graduate student, Tori Arch, who's here, and myself, flew to Borneo because we have a colleague in Brunei, and he lives in the capital city of Bandar Seri Begawan. He's a professor, he's German by birth, but he's a professor now in this University of Biology, and he studies frogs. And we've been friends for a long time, and we've always wanted to do a project together, but not until we discovered this second frog that lives in Borneo, where Ulmar lives also, and we decided to work together and try to find this frog. It's been described, it's in the literature. There are pictures of the tadpole stage and the adult, but nothing about the call and nothing about the behavior whatsoever. And in fact, even the distribution is kind of sketchy. No one really knows where it was, and we had some information which turned out to be false. But then we found somebody who said they once saw one frog in one place, you should try there. So that's what we did. So last year we went to Brunei, and we looked in several places. One place we looked was a field station called Kuala Belalong in Brunei. Spectacular place, and this is where Ulmar works and does his field work. But we didn't find anything there. And what we did was we drove from Bandar Seri Begawan. This is the north coast of Borneo right here. Bandar is up at the top. You can see it up here. We drove in Ulmar's car across the border into Sarawak, which is Malaysian Borneo here. And we went to a little town called Miri, and we flew from Miri up to Muru, the park headquarters. This is the Gunung Mulu National Park, one of the most spectacular parks in the world, as far as I'm concerned. It's beautiful, interesting place, and lots of animals for biologists to study. And from there, we hiked up to a place called Camp 1, the infamous Camp 1. We did this, and you can see here in a more three-dimensional view, this is the airstrip right here. The headquarters of the park is very close. And we hike up this trail up here to Camp 1 at 120 meters next to the Neva River. Okay, we did this with Tori Arch. Here she is arriving herself with me in Muru. In Miri. And we just arrived. Also, this is a picture of Ulmar Grafe one night when we're going to work looking for these frogs. It's a little bit challenging, but it's worth it because we actually did find the frog there. And here's a picture of it. It's called Quia cavitimpanum. Perfect name, cavitimpanum. And this frog also has a tympanic membrane which is recessed and transparent. And in the lower right, you can see a picture of a sound spectrogram of one of its calls. And you can see again the ultrasonic components that it produces. So this frog produces audible sound below the red line and ultrasound, just like the one in China. We were fortunate enough to obtain a device called a recorder made by a company called Sound Devices, and it has a frequency response that goes up to 192 kilohertz, actually a sampling rate of 192 kilohertz. So it's perfectly adequate for recording ultrasound. This is the habitat in the Yippa River. If you take a microphone or a recorder and actually make a recording of the sound of this river, again you can see very similar to the Taohua Creek in China, a very broadband spectrum coming out of the river, perfect kind of masking noise for this frog. This is all very nice because recently a new phylogeny of this whole group came out of Berkeley, University of California, Berkeley, from Brian Stewart, who published this. This is a new phylogeny of this whole group of Narins from Southeast Asia. Up here is the Odorrana livida. This is the Tromota from China, the one we've been studying in China. And down here is the Borneo frog. And you notice they're not very closely related. They're in the same family, but not closely related at all. So what we think is that this is a case of independent evolution, this evolution of ultrasonic sensitivity. It's a case of convergence as a response to parallel ecological pressures. And I'd like to show you a very short video, the last one, for the lights out please, of Huia cavitimpanum in Muru in Sarawak, calling from his natural habitat right next to the Yippa River. And what I'd like you to do is count the number of times you see the vocal sac expanding and contracting. That tells you how many calls he's given. He's going to give. Okay, here we go. So you saw you heard 3 calls, but he actually moved his vocal sac 6 times. That's because in those last 3 calls, he was able to high-pass filter his call. Throw away the audible component. Sorry, it's the other way around. Low-pass filter his call. Tired. Low-pass filter his call, and throw away the audible components and present only ultrasound, which we did not hear. So he's able to modify the spectrum of his call on the fly. This is the first time that any frog is shown to produce pure ultrasonic calls. Okay? It's unusual, it's the first one. So we published this year, and as soon as the paper came out, I received a wonderful email. Have the lights on, please, just for a moment. From a colleague of mine in Chicago called Bob Inger. Bob Inger wrote a book called The Amphibia of Borneo, shown here. And he's kind of the master of these. He spent his whole life studying frogs and amphibians in Borneo. So he saw this paper and he was amazed, and he wrote an email which I'd like to share with you. Can I have the lights off, please? It says this. It says, Peter, I've just seen your paper on the ultrasonic calls of Huia cavitimpanum. Amazing. Equally amazing, they can apparently make themselves heard while they are calling from the banks of noisy rapids. One night, I was standing on the rocky bank of a swift stream in Sarawak and discovered a frog, Odorrana hosii, calling from a twig just about 6 inches from my head. I could hear the frog, but I couldn't communicate with a field assistant who was standing about 3 feet from me. I remember wondering how on earth that frog could communicate with his conspecifics. Your paper solves my puzzle, assuming that Odorrana hosii also has ultrasonic, or at least very high frequency calls. There are a number of species in Borneo that seem nearly restricted to breeding at rapids, where the noise of the stream drowns out human conversation. Do you suppose that those species all have calls with high frequency characteristics? And what does that sunken tympanum have to do with it, Bob? I love this because he had all the insights, he had all the observations, and just didn't have the recorder to make the recordings, but he would have done it if he had that recorder. And finally, I want to thank everybody that has helped us through the years, especially Victoria Toriarch and Marco Scritti-Pop, my postdoc, with work I didn't have a chance to talk about. And Albert Fang, who's been my longtime collaborator from the University of Illinois, Ulmar Grafe, who I've mentioned, Craig Adler at Cornell, who first turned us on to this frog in China. We went to a meeting there and saw this frog and wondered why it had an ear canal. And we've been working on this frog ever since. And all the people that I mentioned earlier from Germany and in the States. Our grant support has been very generous. And speaking of that, I'd like to end with a quote. Because people often ask me, well, are you going to cure cancer or what? You know, what's with you? How come you're not going to cure deafness, or are you? And of course we'd love to, and we have an idea how we can do this, and we're working on this as well. along with Albert Fang. But one nice thing that happened is that when our paper finally did come out in Nature, the director of the National Institute of Deafness and Other Communication Disorders, NIDCD, which is the institute at NIH that funds all our research here at UCLA, Director Jim Batty said this, I quote from him, he said, "The more we can learn about the extraordinary mechanisms that antelopes, that's our frog, and other animals have developed to hear and communicate with one another, the more fully we can understand the hearing process in humans, and the more inspired we can be in developing new treatments for hearing loss." So this is posted on our laboratory, and we say, thanks, Jim. We appreciate this very much, and I thank all of you for sitting through this class.